So this is the last video on translation. We're going to look now at peptide bond formation, the wobble hypothesis, and the effects of mutations. So the process of elongation involves the addition of sequential amino acid residues to the growing peptide chain. The high accuracy of elongation is a function of the specific base pairing between the anticodon of the amino acyl tRNAs and the codon units in the mRNA. And of course, the peptide bond is formed by the ribosomal enzyme. So this again is just a reminder of what that initiation site looks like. We have that Schindel-Garneau sequence where the 16S ribosome binds, and the AUG, or that start codon in the mRNA, in which an FMET is delivered. This slide has diagrams of the important sites in the ribosome. Elongation takes place on the ribosome and is catalyzed by a ribozyme, which is basically a catalytic RNA. The 50S component of the ribosome has 3 binding sites for tRNA. They are designated the A site, which is the amino acyl site, the P site, or the peptidyl site, and the E, or exit site. So the ribosome proceeds along the mRNA in this direction, and the amino acids are brought in as the ribosome moves along. This basically just shows the formation of the first dipeptide in a protein. So with an FMET attached to the P site on the ribosome, as we have here, the next amino acyl tRNA binds to the A site, which is pictured here. And here's our ribosome. The amino acid is delivered to that A site by an elongation factor called EF-TU, and this is a GTP-dependent reaction. So it requires the hydrolysis of GTP to GDP and inorganic phosphate. So that's the energy required for this process to occur. A peptide bond is then formed when the amino group of, and we'll see this reaction in the next slide, amino group, of the amino acyl tRNA at the A site attacks the ester carbonyl group of the FMET tRNA in the P site. And this reaction, again, is catalyzed solely by RNA. At the same time, or following that, we have the translocation of the dipeptide, the FMET being delivered to, we haven't quite, here's the tRNA, I guess. The FMET is transferred to that second amino acid, and then this tRNA will diffuse away. This too is accompanied by an elongation factor G, and requires again GTP hydrolysis. This is an up-close look at that reaction. Here's the FMET tRNA in the P site, the amino acyl, so it's some amino acid in the A site, and that alpha amino group on the incoming amino acid, or the second amino acid, attacks the ester linkage here with the tRNA, or the FMET tRNA in that site. We've already discussed, but just a reminder that the mRNA codons are read 5' to 3', and thus that means the protein is synthesized from the N-terminus to the C-terminus. So termination factors hydrolyze the bond between the protein and tRNA. After hydrolysis severs the link to the tRNA, the protein diffuses away and the ribosomal complex dissociates from the mRNA. So here we have stop codons that are recognized by these release factors, and we've seen these before too: UAG, UAA, UGA, and water is required to hydrolyze that ester attachment of the peptide to that tRNA. This is a look at an antibiotic, puromycin, that is used in cell biology as a selective agent in cell culture systems. It is toxic to both eukaryotic and prokaryotic cells, but it can be used as selection for cells that have acquired an immunity to it. It resembles the 3' end of an aminoacyl tRNA and can bind to the A site, which is, and then is incorporated into the chain, and it causes premature chain release. So instead of a 3', it has a 3' amide linkage instead of the normal ester linkage of the tRNA. So chain elongation terminates. And this diagrams a polyribosome. So this is a number of ribosomes that are translating an mRNA into a protein product, moving along that mRNA. We saw this in this discussion of the location of ribosomes in prokaryotic cells. This is particularly common in prokaryotes, but can happen otherwise too. This is actually electron micrograph of several ribosomes translating the same RNA. So it is a real polyribosome, not just look of it. At this end, they are, you can see the nascent chains are relatively short, and toward this end they're longer, and we are seeing some disassembly of the ribosomes in this region. So now we're going to take a look at the wobble hypothesis, but before we talk about that, I've just put up this slide again. You've seen it before in a previous video that shows the possible 64 codons that code for amino acids, and as you recall, there are several codons that can be read for a certain amino acid. So the wobble hypothesis was proposed by Francis Crick in 1966 and suggests there's some leeway or wobble with the reading of the third base in the codon. The 5' base on the anticodon triplet, which binds to the 3' base on the mRNA, can exhibit non-standard base pairing. So one tRNA can read up to 3 triplets. So we're going to look at some of those unusual base pairings momentarily. So this is an example when a inosine occurs in the 5' end of an anticodon, it can hydrogen bond with a C, A, and a U via 2 hydrogen bonds. And here we see 3 examples of that. We have a CI base pair, an AI base pair, and a UI base pair in that so these are the non-standard base pairings. First base in the anticodon is in this column, third base of the anticodon here, or the codon, excuse me, and possible codes here. So this creates quite a bit of variation in that genetic code. This is just an example of wobble where this tRNA for alanine, so anticodon of 1 tRNA alanine, and these are all recognized then as specific for alanine. And here we have IU, here we have IC, and here we have IA. You've seen this slide before, just to remind you of the process that occurs here. There we go, all of them here. Starting with going from DNA to RNA and to the growing polypeptide, or the pairing of the tRNAs with the amino acids that are coded for by those codons. So template strand, coding strand, our mRNA product, here's our AUG, our start, in which FMET is the first amino acid, followed by the reading of the remaining codons and that growing polypeptide. This is just a slide that shows us another mRNA or DNA. Here's the, actually, no, I believe we're looking at mRNA here. Well, here's the DNA. We've got the TTG-CCA. We have the TA-TT here. Here's a Shine-Dalgarno sequence in the DNA. So this is the promoter region. Whoops. And here's where I'm trying to find here's our ATG. It would be ATG in the DNA sequence. And these are, of course, the amino acids that are corresponding to that. But this is also represents, here we see another Shine-Dalgarno sequence. So this is a region that is coding for many different mRNA products. So now we're going to take a look at mutations, which could be in the form of substitutions or frameshift mutations. So the substitution mutations, and I think I might have gotten those slides a little out of order. Sorry, I might have switched them. Here again, there's the Shine-Dalgarno sequence. Here's our AUG, or our start, and this is in the mRNA. This is the DNA. The ATG is found in the DNA. But the point being here, we're going to look at this codon for alanine. And so this is the normal DNA sequence, the mRNA sequence, and the amino acid order of in the protein or the polypeptide. Here we have a substitution and that substitution is a G here, and this also is read as alanine. So this mutation or substitution mutation is silent because the protein is not affected by that substitution in the codon. We still get the same amino acid as in the normal polypeptide. However, in the third row, we have a missense, or what we call a missense mutation. And so, some of these have little or no effect on protein function, while others may cause profound changes. And in this case, we're substituting a proline, or there is a proline substitution, for an alanine. Frameshift mutations are just what they imply. We have an insertion or a deletion here, which actually shifts the frame. This, again, normal DNA sequence, the mRNA sequence or codons beginning with the start site of translation, or the start of translation. And in this position, we would normally have a phenylalanine. In the insertion here, we have changed that phenylalanine to a tyrosine, and that causes the the insertion causes a stop code to follow that tyrosine. So we would get termination of that chain. So that would be a severely truncated and non-functioning oligopeptide, because it would only have the 4 amino acids. In the third line or region here, we have a deletion. So one of these T's is lost, and the amino acid that is coded now is leucine, which is far different from the aromatic phenylalanine than we have in the original polypeptide. So I'm sure everyone has heard of sickle cell anemia, and this substitution mutation is not benign, and basically, or not basically, it was the first genetic disease to be characterized at a molecular level. And in sickle cell anemia, there is an amino acid chain at position 6 in the beta chain of the hemoglobin. So here is the normal hemoglobin, and in position 6 we have a glutamine, or glutamic, I meant glutamic acid, or glutamate, not glutamine. Sorry about that. We have, with the mutation in this 6-base codon, we now have it coding or reading as a valine. So we've got a mutation from an acidic amino acid to a neutral amino acid. And this dramatically changes the solubility of hemoglobin. And this last slide just shows you a picture of a normal erythrocyte versus a sickle cell erythrocyte. And again, this is all due to that basically a point mutation in that 6th position. These cells will ultimately burst, and the hemoglobin is degraded, and that causes anemia. Well, I hope you've enjoyed translation, and we'll review some of this material next week.